my dear students, how are you all feeling today? I hope all of you are doing great. Aren't you? Are you all energetic? Are you all ready for the EVS class? I can't hear you. Yes. So everyone is ready for the EVS class. We met last week, isn't it so? So today we are going to discuss the remaining topic of the lesson. Okay, so what did we learn in the last week? Let's just recall the topic's name. We learned what all parts of a plant are edible, isn't it so? We learned which all parts of the plant we eat, right? And we learned about photosynthesis, the process by which the plants make food, isn't it so? At that time, I had mentioned some words such as chlorophyll, stomata. Do you remember? So today we will discuss in detail about all this. We'll discuss about how plants get carbon dioxide. We will see the experiments. So are you ready? Yes, right? And then have you ever thought, ma'am told the chlorophyll, the green color in the chlorophyll gives the leaves green in color. Then how was some yellow and red colored leaves there? Have you seen the yellow and red colored leaves? Yes, right? So today we are going to discuss how it is not green but yellow and red. So are you ready, my dear children? Yes. So then let's go. So my dear children, shall we start? Yes. The first, let me ask you a small question. We discussed it last week. From where do plants get carbon dioxide? From us, right? From the animals. How do the plants get the carbon dioxide? How does the plant absorb the carbon dioxide? We talked about stomata last week. Stomata helps in taking the carbon dioxide gas from the air, right? So, today at the beginning, let's see a small experiment where we will observe the stomata through a microscope. That is, we will observe the leaf and see the stomata. Are you ready? After that, we'll come back and read the text and see how the activity is given here. Are you ready? Yes.
To prepare a temporary mount of a leaf peel to show its stomata. Materials required: fresh leaf of a lily plant, slide, watch glass, forceps, glycerine, brush, pair of sharp scissors, saffron in solution, blotting paper, cover slip, compound microscope. Procedure: hold a freshly plucked lily leaf. Break the leaf from front to back by putting suitable pressure so that the peel of lower surface comes out. Ensure that you are taking the epidermis from the lower side as the dorsal surface is rich in stomata. Cut small pieces of the peel into watch glass filled with water. Add a few drops of saffronin into the watch glass to stain the leaf peel. After 2 minutes, place the stained peel in water in the watch glass again to wash off extra stain. Put a drop of glycerine on the slide. With the help of a brush, transfer a piece of peel on the slide. Put the cover slip over the peel gently by first making an angle of 45 degrees with the slide with the help of forceps. The peel should never be folded. Place the cover slip on the slide gently and gradually to avoid the entry of air bubbles. Drain out the excess stain by blotting paper. Examine under the low power of the microscope. Observations. The epidermis is made of uniseriate layers of cells. The cells are closely packed together with distinct cell walls. Each stomata is guarded by a pair of bean-shaped cells called guard cells. Guard cells are rich in chloroplasts.
So everyone watched the experiment, right? You saw the stomata. We have stomatas in all your leaves, but we cannot see them with our eyes, right? Okay, so then let's start reading the textbook. Peel off a little of the outer layer of the lower side of a betel leaf or a taro colossia leaf. Dip the layer in dilute stain. Place this layer on a glass slide using a point brush. Observe this through a microscope. Don't you see pores in the leaves? What we did in this experiment which ma'am gave was we used saffronin, and saffronin is that dilute stain which helps us to see stomata. Here the experiment is given in a very simple way, but we get the same results while doing like this or the way ma'am showed you. Is it clear, my dears? Yes, right? Good. Now let us see the definition of the stomata. Right. Plants absorb carbon dioxide and release oxygen during photosynthesis. All of us know that. We learned photosynthesis has a product which gives out oxygen by taking in carbon dioxide. Right, we learned it last week. Wasn't that clear for you? Now, this gaseous exchange, that is the exchange of gases, takes place through small pores in the leaves. Which are the small pores in the leaves? Just now we saw in the experiment, which was the small pores. These pores are called the stomata. They also release water from plants to the atmosphere. When the plant's roots absorb the water, when there is a lot of water in the plant, the excess water is removed through the stomata in the leaf. Is it clear? Now can ma'am give you a small activity? Take your science book and draw the stomata in your book. Okay? Yes, good children. Now, observe the plants around you. Go to the garden once your class is over. What will you see? How does the plants stay? Like which part of the plant is more exposed to sunlight? Like which part of the plant does the sunlight fall more on? Where does the sunlight fall more on a plant? You have seen it before. The plant is full of leaves. We have seen mostly the sunlight is trapped on the leaves, right? The sunlight falls directly on the leaves of the plants. Isn't that so? Yes. So now we are going to learn that little secret which gives all the leaves green color. What did we say last week? It was the chlorophyll which gave the leaves green color, right? The green colored pigment seen in plants is called the chlorophyll. What does chlorophyll do? Chlorophyll absorbs the sunlight needed for photosynthesis. Chlorophyll is present more in the leaves. In a plant, the chlorophyll is seen more in the leaves. And what does this chlorophyll do? It's a green pigment, so it gives a green color to the leaves, as well as it also traps the sunlight which is required for photosynthesis. Now, all leaves are green or not? What do you say? Have you seen only green leaves? If you ask me, I would say not all leaves are green. You also know that not all leaves are green, right? Then what is that secret which changes the color from green to red or yellow? Have you seen the red amaranthus, which is chuvanna cheera in Malayalam, right? Have you seen that? All of you must have eaten also. Isn't it so? Okay, so then again there is an experiment here. We'll read it out, okay? But what is the aim of the experiment? Before doing any experiment, we must know an aim, isn't it so? So what is the aim of the experiment? It is to find out whether chlorophyll is present in the red amaranthus too. It is not green in color. Then is chlorophyll present or not? Let us see. What are the materials required for the activity? We need leaf of the red amaranthus and blotting paper. What we should do? Rub the leaf of red amaranthus on a blotting paper. Observe the color change. Observe the color change in the paper. Do you know what it will turn out? Can you guess which color it will turn out? It is not red, it is a greenish color. So what does that mean? It means that there is chlorophyll or the green pigment present in red amaranthus too. Then how is red amaranthus red in color? How are the leaves red there? All other leaves also have chlorophyll and they are green in color. Then why only red amaranthus? Not only red amaranthus, all the leaves with red and yellow are in different colors. There is chlorophyll, then how are they in different colors? Now we will discuss about that. Are you ready? Are you listening to me clearly? I can't hear you. Yes, all of you are good children, right? So again, we are going to break out a secret. How is the colors different from leaf to leaf? Some are red leaves, yellow leaves and green leaves. And all these yellow and red leaves also have chlorophyll. We saw that in the last activity, right? So now, chlorophyll is a pigment, right? And the pigment's color is green. Which color is chlorophyll? It is green. But there are different pigments also present in a plant. That is why when a different pigment other than chlorophyll is more in quantity in a plant, then it appears to be of different color. For example, if it is xanthophyll pigment, it is called xanthophyll. You have to write this down in your diary, in your notebook. Xanthophyll. Xanthophyll spelling is XANTHOPHYLL. Xanthophyll. And xanthophyll pigment gives yellow color to the leaves. Then what about those appear in yellowish orange? Not only yellow but orange also slightly. That is called the carotene. 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 This gives the yellowish orange color. What about the red color for the red amaranthus? That pigment is called anthocyanin. ANTHOCYANIN. Okay. So in red amaranthus, anthocyanin pigment is there more than the chlorophyll pigment. So red color is more, and that is why we see a red color leaf. Clear? So what did we say? We had carotene for yellowish orange, xanthophyll for yellowish leaves, and anthocyanin for red colored leaves. Now, have you ever thought that are pigments present only in leaves? Chlorophyll is a pigment, xanthophyll is a pigment, the anthocyanin, the carotene, all are pigments. Are they present only in leaves? Then what is the color? Why is hibiscus at times red? Why is it white at times? And why is the sunflower yellow? Right? So pigments does not only give color to the leaves, but also it gives color to the leaves, stems, flowers, and also fruits. Clear? Okay. How many of you have plants inside your home? Do you like keeping plants inside your home? Why are you keeping it? What for? Is it for decoration? For us to see it would be beautiful inside the house? Now see, a little boy named Baiju said to his teacher, "Teacher, I have grown some plants in my bedroom. Won't there be enough oxygen released by the plants inside the house?" He asked this question to his teacher. Now Baiju asked this to his teacher. But I want to ask it to you. Is there any benefit growing plants inside the house? Is it only for the decorative idea? Is it so? No. It has a lot of other uses. Okay? It cleans the air inside the house. It increases the humidity. It increases the oxygen level in the house. Right? Yes, it does. Then will this arrangement help us get oxygen in the night? Most of the indoor plants absorb CO2, absorb the carbon dioxide in the night and release oxygen. But the outdoor plants mostly only absorb carbon dioxide during the day and release oxygen. Right? Isn't that so? So my dears, that was for today. It was a very small topic, isn't it so? I hope you understood the lesson, you enjoyed the lesson. What did we learn about? We learned about the stomata, the chlorophyll and the pigments which change the color in the leaves, right? So if you like my class, then please let me know in the comment box below. And if you have any doubts, clear it in the discussion. So we'll see you in the next video in the next week. Till then, bye bye, take care and stay safe, dear children.